Hello and welcome back to revise GCSE history.co.uk. This is the fourth video in the topic Medicine in the 19th century, and in this video today we're going to be looking at the work of two individuals, and they are Louis Pasteur and Robert Koch. We can sometimes pronounce Robert Koch as Robert Koch. People pronounce it differently and it doesn't matter, either one you can use. So these two individuals, that's who we're going to be looking at today. So, before we begin to look at these individuals, let's have a look at this. What did people think caused disease at the start of the 19th century? Obviously, people didn't know that germs cause disease, and that's what makes people ill. But here are some things they did think made people ill. Miasma, they thought disease was caused by bad air. There were poisonous fumes in the air given off by rubbish and decaying matter. Contagion, they understood this, and this is certainly true. That's being close to someone with the disease meant that you got the disease too, and we know that's true today. God, people still believe that disease was a punishment from God, as they did in the Middle Ages. And later on in the 19th century, they believed the theory of spontaneous generation, which was that decay creates germs, and that germs appear as a result of disease. This is sort of the opposite to the germ theory we understand today, that germs cause disease. Spontaneous generation is that disease causes germs. So let's have a look at the first individual, which is Louis Pasteur. And these two individuals we're going to be looking at in today's video are important in the fight against infectious disease. Okay, so Pasteur. Microbes have been seen through 18th century microscopes, but scientists thought they were caused by disease, and appeared because of illness. This was the theory of spontaneous generation, just been talking about that. So it's sort of the opposite in terms of what we believe today. They thought that disease causes germs. However, today we know that germs cause disease. Instead of blaming microbes for disease, people look for gases in the air called miasmas. They thought this made people ill. Louis Pasteur was employed in 1857 to find the explanation for the souring of sugar beet used in the fermenting of alcohol. So souring of sugar beet, and he said that it's going sour because there are germs in the air. So this was a new theory. So Louis Pasteur was employed by a brewery to find out why the sugar beet used in the production of alcohol was going sour. Pasteur proved that there were germs in the air, by sterilising some water and keeping it in flasks that didn't allow airborne particles to enter. This is called the swan neck flask experiment. So takes some water and puts it in a flask that doesn't allow airborne particles to enter. We call it airtight. The water in the airtight flask that was shut off to the air stayed sterile, but the sterilised water kept in an open flask, one which could be come into contact with the air, bred microbes. So that proved that there were germs in the air. And Pasteur published what we call his germ theory. We're going to be looking at this a bit more in a sec. So he published his germ theory in 1861. There is a bit of discrepancy over the date that germ theory was published. I've always understood it was published in 1861. Some say it was published in 1857, but I think that's when he was employed by the brewery. 1861, you'll be fine with that. Whoops, sorry about that. There we are. So what is spontaneous generation and what is germ theory? Here's just a diagram that illustrates that. So they say that microbes are the result of decay. Decaying matter itself turns into living organisms. Early versions of the theory suggested that even the flies and maggots that were seen on decaying matter were created by it. Here's a diagram that shows that. But this is a germ theory, and this is what Louis Pasteur said. That's Louis Pasteur. That's what he said in 18 1861, and this is before Pasteur, and we still accept germ theory today as it is scientifically correct. So germ theory is that living organisms fall from the air and cause decay, and decay causes illness. So that is a quick diagram that illustrates that. Next thing, Louis Pasteur's germ theory, we've already spoke about this. It was published in 1861, and in 1864 he devised a series of public experiments to prove that his theories were correct. So his work in 1864 helped prove to scientists that his work was correct, as he demonstrated this in a public experiment. So Pasteur thought that disease could be caused by the same process as wine going sour or material decaying. He proved that microbes seemed to be causing the silkworm's disease, as he was asked to investigate this by the silk industry. So here are some of the experiments he did and the theories he took them from. So Pasteur said that the air contains living microorganisms. He needed to prove this, so what he did, he took sterile flasks out into the streets of Paris, opened them briefly, then sealed them again. Bacteria grew in them. Just talked about that really, how germs are present in the air. Another theory he had is that microbes are not evenly distributed in the air. So what he did to prove this, he repeated the experiment in various places around France, including high mountains. The number of bacteria in the air varied. Another theory he had was that microbes cause decay, and what he did to prove this, he filled two flasks with water, one with sterile air and the other with ordinary air. In the first there was no decay, in the second decay proceeded as normal. So that's microbes in the air causing decay. Microbes can be killed by heating, or today we call this pasteurizing. He heated material in a flask to make it sterile. He drove the air out, then sealed the flask. It remained sterile even 100 years later. So microbes can be killed by heating. So why is Louis Pasteur significant in the history of medicine? This stuff is what you need to know for a question that asks you why is an individual important in the history of medicine. You need to assess the significance of them. So that he proved that germs cause matter to decay, spontaneous generation proven wrong, disease is not caused by miasma, people thought it was at the time. Set up a series of public experiments in 1864 to show that germs cause decay and that sterile air does not decay. Linked decay caused by germs to the cause of disease. He realised that germs cause disease, basically. This is why it's important in the history of medicine, because before Pasteur we had all these theories, such as the theory of the four humours, miasma, punishment from God, all this sort of stuff, and none of that was true, and none of that is really what causes disease. Pasteur's germ theory is the true cause of disease, and the cause of disease that we understand and accept today. So for the first time, people were right about what was causing disease, and treatments could be developed from this. So that's the stuff we need to know about the first individual, Louis Pasteur. Here's some stuff on the second individual now, Robert Koch, or Robert Koch. So, this is some stuff he did on identifying bacteria. So Robert Koch was a German doctor. He developed an interest in Louis Pasteur's germ theory, which was published in 1861. So Louis Pasteur sort of set Koch's work off in motion, developed an interest in that and thought, I want to do something like that. He investigated anthrax, which is a disease affecting animals and humans. It's quite common in sheep. In 1875 he managed to discover the specific bacterium that causes anthrax. This was the first time the specific germ that caused a specific disease had been identified, and it was the final proof of Pasteur's germ theory. So what Koch started to do is link specific diseases to the specific microbe that caused them. He understood that each disease is caused by a specific microbe, and he did this when he did so for anthrax. Koch also identified the bacteria that cause septicemia, tuberculosis and cholera. Koch also developed a method of proving which bacterium was causing a disease through his experiments with his daughter's pet mice. That's right, he did experiments on mice to prove which particular bacterium was causing disease. So that here, this slide is giving information about Robert Koch's work on identifying bacteria. He also did some work on studying bacteria. Let's have a look at this. So, Koch developed a solid medium as an easier way to study bacteria. Before the solid medium, people used to use liquid mediums to study bacteria, and Koch thought it was easier to use a solid medium. He actually grew his bacteria on potatoes. That's why he used a solid medium. He also developed new ways of staining the bacteria. He stained them using a purple colouring so they could be seen more easily. Robert Koch also developed ways of staining bacteria so they could be photographed using a new high quality new high quality photographic lens, and then studied in further detail. So developed ways of staining bacteria so they could be photographed. So that is some key points about Robert Koch's work in studying bacteria. So, what have we got so far in terms of bacteriology? We've got 3 key individuals. We've only talked about 2 individuals in this video, but you will remember one individual from earlier on in the 19th century medicine. So the first individual was Edward Jenner. We spoke about him in video 2 of this topic. He discovered that cowpox vaccinates people against smallpox. So they have cowpox, which is a milder disease than smallpox, so they won't catch the smallpox disease. However, he doesn't know what causes smallpox or how vaccination actually works, so a bit more work needs to be done in that area. The second individual is Louis Pasteur. He discovers that germs cause decay, they make things rot. He develops his germ theory, and he links germs with disease. However, Koch does not identify specific germs with the disease. Sorry, I've made a key mistake there. I've said the wrong thing. It's all right on the slides, what I'm saying is wrong. Pasteur does not identify specific germs with specific diseases. Robert Koch later does this. So Robert Koch, individual 3, he identifies microbes which cause a specific disease, for example TB, cholera, septicemia. He develops new ways of identifying bacteria, solid medium, petri dishes, photography techniques and dyeing techniques. However, another drawback, he does not develop cures to kill these bacteria. So, in chronological order, we've got Jenner, Pasteur and Koch, and they are the 3 individuals making progress in the field of bacteriology. So, we've looked so far in this video at Pasteur and Koch, but there's a little bit more we need to look at now, and that's what happens next. So we've got this thing happening on this slide. What happens after all this? So, Louis Pasteur hears of Koch's work and comes out of retirement in 1877, and he starts to compete in the race to find new microbes and combat them. Many other scientists joined this new field of bacteriology. Pasteur looked for cures to anthrax and chicken cholera. Anthrax is a disease which affects mainly sheep, and chicken cholera affects chickens, quite obviously. Both Pasteur and Koch, remember he's French, he's German, worked with large teams of scientists in this Franco-German competition for national... Sorry, can't talk today. In this Franco-German competition for national prestige. So it's the French versus the Germans in this fight to find new microbes and combat them. Charles Chamberlain was in Pasteur's team. Charles Chamberlain, you could pronounce it. That's how the French pronounce it. And we'll look at more about him right now. So, the chicken cholera vaccine was discovered by Louis Pasteur due to the mistakes of Charles Chamberlain. So one day Chamberlain was told to inject some chickens with chicken cholera, but it was the day before his holiday and he forgot. Silly Chamberlain. Chamberlain left the chicken cholera germs on his desk and injected the chickens when he returned from his holiday, say a couple of days later. However, the chickens survived. What am I on about? The chickens survived because chicken cholera was quite a deadly disease. It would usually kill chickens. Chamberlain was quite surprised when he injected these chickens and they actually survived. They tried again with some newly cultured cholera germs, but the chickens still survived, even against the new germs. Pasteur and Chamberlain worked out that the cholera had been weakened, the original germs had been weakened by being left on the desk for a few days, and that the weakened cholera germs that were left on the desk while Chamberlain went on holiday had made the chickens immune to chicken cholera. This meant they wouldn't catch it ever again. So this was in the same way that Jenner's cowpox vaccine had worked for smallpox. Chamberlain's error had made a chance discovery. So that's a great example of chance affecting the development of medicine. So basically, why that works and how it works is Chamberlain left out the germs on the desk. They were left there for a couple of days. Being open to the air, the germs weakened, and the weakened version of the chicken cholera disease had made the chickens immune to the normal chicken cholera germs when they were injected with some new germs. So that is a vaccination that was produced by Louis Pasteur and Charles Chamberlain. There are also further vaccinations from Louis Pasteur. Let's remember he's French. So Pasteur's team managed to produce a weakened version of the anthrax spore that would make sheep immune to the disease. They demonstrated this in a public experiment in 1881. That's another thing you need to be aware of. So, that's all the knowledge we need to cover for this video today. Here are 4 quick questions based on the content of today's video. You may wish to pause the video here, jot down a few answers. If not, carry on watching, we'll do them together. Okay, so question 1. What 4 things did people think caused disease at the start of the 19th century? We've got miasma, name all 4 here. Contagion, being too close to someone with a disease would give you it. They still believed disease was a punishment from God. And also, they believed in spontaneous generation. Oops, a bit untidy. Spontaneous generation. They're the 4 things. Describe the idea of Pasteur's germ theory. He said, germs are in the air, and they cause decay. He then said, if germs are causing decay, do they cause disease? That's what he said. Jot down an answer in note form, hopefully you can understand that. Next question to look at now. Okay, so give 2 techniques developed by Robert Koch and for studying bacteria. He developed a solid medium. Remember, we use potatoes. He developed a staining or dyeing technique. Staining technique. Okay, final question for this video today. What factor led to the discovery of the chicken cholera vaccine? And I would say that chance is the most important factor there. Alright, so thank you for watching this video today on the work of Louis Pasteur and Robert Koch. Next up in this series is a video on surgery in the 1800s. Thanks for watching, see you soon.